Thank you.
Yeah, one of the things we wanted to do that would be a little different, I don't know if any of you have been to our previous annual meetings, but we decided we wanted to do something a little bit more interactive and hopefully interesting. So with the help of Alan Kugel, who's on our board, and this is his home turf, he's from the Pew Charitable Trust, we're taking a shot at looking at big data and our project IMEDS, and we're going to have a discussion with a number of stakeholders. But at this point, I'll hand it over to Alan to introduce the panel.
Thanks, Jane, and good morning. So I'm Alan Cockle, and I direct health programs at Pew, but more importantly, for purposes of today, I'm a board member at Reagan-Udall. And as Jane said, at a recent board meeting, we had a discussion about what could we do to use this open public meeting to more effectively explain the work of the foundation to interested stakeholders. And one of the ideas that came out of that conversation was that these days, every time you turn around, you read an article or see a panel about big data. The potential of big data is everywhere. But a lot of those presentations sort of jump from the potential of big data to the results, and they don't explain all of the hard work in between and all of the things you have to do to make big data usable. And Reagan-Udall Foundation is doing that with the IMEDS program. IMEDS is Innovation in Medical Evidence Development and Surveillance, and it's the largest initiative of Reagan-Udall. And so we have a distinguished panel here, both to talk about big data in general, but also to use IMEDS as kind of a lens to talk about what does it take to make big data work. So you have the detailed bios for the panel in your packets or on the table at the back. So I'm not going to do long introductions of everyone here, but just briefly, to my left is Marcus Wilson, who's president of Healthcore, which is Anthem's outcomes research subsidiary. And he's also the chair of the IMED steering committee. To his left, Sally Okun is vice president for advocacy at PatientsLikeMe and a member of the scientific advisory committee for IMEDS. Bob Ball is Deputy Director for the Office of Surveillance and Epidemiology at FDA, and he has a long history at FDA, which has included a lot of involvement with the Sentinel program, which is very relevant for today's discussion. And to his left, Troy McCall, who's IMED's Chief Implementation Officer. So I'm really glad to have all of them here. And so let me start with a kind of open-ended question to each of you, and we'll just go down the line, and we'll get to IMEDS. But start more generally with where we stand now in our use of big data and the vision that each of you have for where we'd like to go.
Thanks, Alan. It's a pretty open-ended question. You can go a lot of different ways with that. So I'm glad to be here. Thanks for Jane and the crew for having us here, and Alan for organizing the panel. So it's interesting. I'll start with this, and we can kind of fill it in as we go with this as a group. But being part of a large payer, or a subsidiary of a large payer, Anthem in this case, you get a chance to get a view of healthcare from the payer side of things. And Healthcore it's a company we started back in 1996, and we sold it to Anthem in 2003. At that time, it was WellPoint, and the reason we did that is because in working for the previous 10 or 15 years in my career to that, we'd tried looking at different types of data and different ways to construct a better view of the individual and ultimately of populations, and with the idea that we could do two things with it. And one of them is generate better insight. And then the other is we could sort of use that same framework to, once we've generated the insight, to sort of quickly get that into practice and support decision making. So ultimately, we're trying to make better decisions. And what's interesting in all this, if you think about a health plan, interesting to me anyway, but as a health plan, the health plan has this concept that many of you may know well, and it's called gaps in care. And we spend enormous amounts of money, not just us, but the healthcare system as a whole, trying to close gaps in care. And a gap in care is just by definition, very simplistically put, it's that trying to get the physicians and the providers to do what they should be doing. And so you're looking for when variances to those treatment patterns and that you think should be happening, and you say, look, you shouldn't be doing that, you should be doing this. But here's an interesting dilemma in all of this, and it's framed out very well. We had Rob Califf is on our steering committee as well, and about a year and a half ago at one of our board meetings, he quoted a piece of evidence that I now use quite a bit, and it was some work that he had and his team had done back in 2009, I think, as published in JAMA, and they looked at the quality of evidence that goes into guidelines. Now, remember, gaps in care, as I said before, is we should be doing this, and here's what you're doing. So if there's a difference, then we consider that a gap, and we throw all types of nurses and physicians and tools and analytic methods and everything towards these populations to get them to do what they should be doing. And the basis for those is guidelines, right? Those are treatment guidelines are really the basis of the framework. It's a framework for HEDIS. It's a framework for all these quality measures. Now we're doing it in performance measurement for physicians. Long-winded way to get to one thing on this. I really don't think gaps in care are the issue. I think there's a bigger underlying issue in all of that. And that is really, as I go back to Rob's article that he quoted, when they looked at the various cardiovascular guidelines out there that graded the evidence that went in the guidelines, they took 16, now they distilled it down to 16 major guidelines that governed all types of, or guided all types of care and decision-making in cardiovascular disease. And of the 2,700 recommendations inside of those 16 guidelines, only 11% had sufficient evidence to support the recommendation. The majority of that's based upon expert opinion. Now, there's other things, but expert opinion is a big part of this. And what's the problem with expert opinion? Right? Every expert has a different opinion. And it's not necessarily ill-founded. In many cases, it's very, very well-founded. And the challenge is that what guidelines sometimes don't have the granularity to do, and what's missing in a lot of cases is really what do we do with that individual sitting in front of us. And that person is probably very different than the evidence that was generated on populations that were, so the exposure is different than the ones that are studied in real world settings. So for us, what we look at this is we started out looking on generating new evidence based upon the ability to close a gap in knowledge or gaps in evidence. So instead of gap in care, what we've really started to focus on a lot, and we've been trying to do with pulling all those data together over time to create the better view, is to close the gap in evidence. And then we can close the gap in knowledge much more effectively. So I think for me, in our group, a lot of our effort, and I think the IMEDS program is very much based on that philosophical foundation that it's critical that we find better, more efficient, more rapid ways to generate relevant evidence to populations that the decisions are applied to. And I think we're going down the pathway. I think we've got a long ways to go. I think there's still a lot to learn about the data that we're repurposing for this, but I think IMEDS is very much committed to doing it. Starting with a foundation of safety, we're really trying to leverage that into other types of outcomes beyond just safety and time as well. So I'll stop with that and turn it over to the rest of the panel.
So Sally, let me go to you. So what's your vision of how we can use data in ways that we're not yet using it?
Well, thank you. I think the interesting piece that I can follow on here that I think is very relevant to the work we do at Patients Like Me, and for those of you who are unfamiliar with it, is a web-based environment for patients to create their own health profiles, to monitor their treatments and their symptoms and their conditions. We are able to then aggregate and make some insights from that data. So when we started that 10 years ago, the notion of patient-generated data was really very unheard of. But today, it's almost ubiquitous. You hear that term all the time. But we still also hear, what's the role of patient-generated health data in big data? I've heard it called small data to distinguish it, which always sort of surprises me a little bit. But what I would suggest is that actually, as we continue to better understand the nuances and the variety of information we can gather from individuals who are proactively and quantitatively gathering information about their own experiences, we have a treasure trove of information that we haven't even begun to think about how to use. I often say that actually patient-generated data is the ground truth that's been missing from big data for too long, and that the opportunity today is to think about how do we start thinking about the methods needed to better understand it. And I think that's where IMEDS and the Reagan-Udall Foundation has really stood out They recognized early on, first of all, by even inviting me to be on the scientific advisory committee, but also as part of the research agenda, understanding that novel data sources will continue to emerge, and we need to figure out how to use them and to answer important questions, as we've already said, at the point of care, when that individual is sitting there and really needs to think about, well, what do other people like me experience? How is my situation unique? So again, thinking about the fact that big data, when it's generally defined, it's got a lot of volume, it's happening very quickly, it's got a lot of variety, but we're still a little uncertain about the veracity. And I think I really would put the call out there to say that as we circle back around and we start to add the patient voice in a quantifiable way, even in a qualitative way, beginning to think about that, test our assumptions about what's true and what's not by bringing that back into the bedside to bench model instead of thinking always from the bench to bedside. I think we can continue to move that from bedside to bench to move knowledge from one place to another and then back to the bedside again in a very effective way when we start thinking about the use of real-world evidence, real-world data, and the engagement of real people in the involvement in their health to understand how data can actually make a difference in the decisions that they'll be making. So I'm thrilled to be a part of IMEDS and see its progression over time and start to think more concretely, what is the role of patient-generated health data, and how do we together start to figure that out in ways that we can test some of the methods within the IMEDS laboratory and other ways to really tease out the contribution, this very rich, diverse, and somewhat messy data. Real-world data is definitely messy, but we will have the opportunity, I think, with the work that's being done here to really start to tease out the best ways of being able to use it, gain insights from it, understand the value it brings, and then act on it and make some real use of it in the healthcare ecosystem.
So, Bob, FDA uses data every day, obviously lots and lots of prospectively collected data, but also lots and lots of observational data from a variety of sources, including increasingly the Mini Sentinel program. So as you look at where we are now, what's the vision of what more we can do, where we want to get to?
So I have to admit, when I first started hearing about big data, a number of years ago, I asked myself the question, how is this different than what we do now with large-scale data? So we have for many years used CMS data, claims data to do safety evaluations. And so my first order of business, I guess, was to think about, as a clinician and epidemiologist, how can I reframe this concept of big data to fit into what I do every day? So I came up with I guess, 3 concepts which I've extracted from various places. So this really isn't original. But I think of it in this way. I think of bigness, meaning that we have access to large numbers of people, which lets us look at very rare conditions and also the whole diversity of clinical conditions. We also have access to very thick data. So thickness in this context means that on each one of those people, we have lots of different types of data and large quantities of it. And this becomes very important as we try to make inferences that are scientifically valid. And the third piece of big data is what I think of as the diversity of the data. So we started hearing a little bit about that. There's patient-generated data, there's claims data. We also know there's genomic data, there is mechanistic data on how drugs actually work. And all this data is out there circulating. The question really is, I think, for big data, is how do we bring all this data together to make inferences in a scientifically rigorous way about whether it's drug safety or in the future, hopefully, drug effectiveness in ways that we haven't been able to do in the past.
Great. So Troy, I want to begin shifting us to talking about IMEDS, but could I ask you just to give, after this session, you're going to give the formal Reagan-Udall update on the IMEDS program, but just for purpose of this discussion, a quick kind of, in a nutshell, overview of IMEDS. What's the data that we're looking at? Where's it coming from? Who owns it?
Sure. So I think I'll start out by answering that question, Alan, by talking about why was IMEDS formed in the first place. And looking back in time, there were three really important gaps in safety surveillance of marketed medical products and medical devices. And those gaps were, one, there really was not a central home whereby methods were being developed that really took into consideration all of the different thoughts and ideas and perspectives of different stakeholder groups, such as FDA, industry, academia, patient advocates, and so forth. So that's one key area. We've created a central way in which methods can be facilitated. The second area is this is an emerging science. This is, relatively speaking, in its infancy stages. And as a result of that, we need to expand the scientific community and the community of researchers that are capable of conducting this type of research. So we need to create an environment to enhance the educational opportunities. And then the third is, there has been, and we'll talk about this during the formal presentation, but no opportunity for non-FDA stakeholders to access the tools that exist within Sentinel, the methods, and importantly, the data sets represented by the 18 data partners. So the third component of IMEDS is IMEDS evaluation, and that will present an opportunity for non-FDA stakeholders to access those methods, tools, and data sets. Of course, FDA is the owner of Sentinel and will always be the one to access Sentinel. But we need the opportunity to be able to assess the safety by non-FDA stakeholders, such as industry, for example.
And so the data that IMEDS has is similar to what Sentinel has. Just say a couple of words about that.
Yeah, so there are two kind of ways in which we look at data within IMEDS. The first is within IMEDS methods. There we have a database, a group of databases, data sets that reside on the cloud. We actually access data from 5 different data sets that represent over 80 million lives. And when we talk about that residing within the IMEDS research laboratory, I think a lot of people think of a laboratory and you think of people sitting at a bench in white lab coats. Do not think of this laboratory in that perspective. This is a virtual laboratory where all the data sits on the cloud and researchers are able to go in and access all those 80, lots and lots of data, as we've talked about big data, in order to develop new methods, in order to refine existing methods so that we can eventually come out with the best methods that FDA, industry, and others can apply to big data sets. The second area of data is represented within the IMEDS evaluation program, and that's where we are developing relationships with each of the 18 data partners such that we can access their distributed data network. So IMEDS methods is not distributed, it resides on the cloud. IMEDS evaluation is where we have the distributed data network that we can actually ask queries of the system.
So, Bob, let me go to you. From FDA's perspective, what's the complementarity between IMEDS and Sentinel? How do they work together?
Well, I think to follow up on what Troy was saying, a couple of years ago, the Observational Medical Outcomes Partnership, which was a research consortium, did some methods development work that I think was in some ways a surprise to the whole field of epidemiology and pharmacoepidemiology and drug safety. And one of the main points of research for the IMEDS activities is to better understand those OMOP findings and to help us understand how they apply to the Sentinel initiative. And I think that's like the perfect example because it's a group that's external to FDA but focusing on FDA's specific needs. And going forward in this year's research agenda, similarly, there's the Sentinel group has developed a number of new methods. And one of the goals of the scientific agenda is for third parties to actually evaluate those methods in data that's outside of Sentinel and define their performance characteristics. So that's a very concrete and very useful for us as the FDA to understand actually how these methods perform. And I think going forward, this whole idea of a place where methods can be developed, tested, and then implemented is really critical for how FDA can transparently further this whole field.
So you've both used the words methods a lot, and I'm not sure that's totally obvious to those of us who aren't in the field. So I wonder if there's an example. You mentioned the OMOP findings. I wonder if that or something else would offer an example of what does it mean to develop methods to be able to make sense of these data? Either one of you.
Okay, so I think that just to get very concrete about it, and when we look at drug safety, we ask the question, did this drug cause this clinical outcome? And when we look at an individual patient, we generally aren't able to do that in a scientifically rigorous way. So what we do then is we say, let me compare within a population how people who got the drug and people who didn't get the drug, how, whether or not they had similar kinds of clinical outcomes. And at a that's sort of the simple level. But as soon as you start getting into this, you realize that there's tremendous complexities because the patients that you're comparing aren't the same. Many people have many different underlying medical conditions. The drugs they may be taking for many different conditions may also influence the outcomes you're looking at. And how one then tries to study those questions in millions of patients with thousands of drugs and thousands of outcomes is really what methods development is about. And it's so that we can do this in a rigorous way that, so when we say we think this drug causes this condition, we can be confident in that conclusion.
Do you want to jump in here?
So just to complement what Bob just said on this is that, so that there's the methods, that the techniques you use to analyze the data, to whether that's statistical or more broadly defined. The other aspect of sort of the methods development that from my perspective is also really trying to improve the quality or enhance the quality of the data underlying it. So you can the sort of the mantra out there around claims data is if you torture it long enough, it'll tell you what you want it to say, and it's very true. And so what we're trying to do in terms of getting data itself, the underlying data, much more reliable, is to get it much more consistent and make sure we understand when it varies, what's really leading to that variance. So in Bob's example, so if we look at this population versus another population, and we see the presence of an adverse event in this population, but not this, before we sort of attribute it to a pharmaceutical or biotechnology or any other cause, we want to make sure that there's not something underlying in the data and the populations that's different. So if there's differences in the populations that we can see, we can adjust for that. There's good methods for adjusting for difference that you can see. The challenge is that what if there's variations that are not obvious in the data? So if you take, as an example, claims data, and you take a population that comes from an Anthem health plan in diabetes, one of the things that the data won't tell you is is there a difference in the populations when it comes to the care management programs with which they've been involved. So if we had a lot of those nurses I've referred to before working with more of the patients in one cohort trying to improve quality of care and outcomes than the other, that's an underlying confounding factor you would need to adjust for when you're doing your matching. And so we can't, one of the really important lessons about data of all types that we use that are originally for other purposes is to make sure that we have good ways to go back and deconfound the data before we draw too definitive conclusions from it. So as we're putting these things together, as Sally talked about, putting different types of data together, which is imperative, that we do it with care. And I look at a lot of the work in Sentinel and how Sentinel was constructed. We've been part of it since its origins. Is that it's being put together very thoughtfully, very carefully, so that when we need to, we can go back and look into that. And a lot of what we're taking into IMEDS and complementing the work in Sentinel is really aimed at the same type of mentality. And honestly, a big reason why I'm I agreed to be a part of IMEDS in the first place is because that desire to make sure that we continue to look at the same type of discipline, and the group in IMEDS is certainly doing that. So.
I think I'd like to just give an example, too, from a real-world evidence perspective. We actually worked with one of our pharmaceutical partners about a year or so ago on just conducting a pilot to see if we were to take data about a couple of drugs in our system from the reports of patients, and then sort of ran through the signal detection methodologies that are traditionally used. How might that, what would the results start to look like? And it was interesting because our partners, our colleagues, were a bit skeptical. They were sort of just wanted to test this and kind of see what they would find. And they were delighted, actually, at the end of the pilot to determine that the standard methodologies that they would use in any sort of data source could be used in our data. In fact, what they found was there were a couple of things that they hadn't seen before in both of these drugs. And then the next step was to say, okay, now we need to think about how to validate that, how to take those findings and not necessarily believe them on face value, but dig deeper and understand what is the nuance within that actually we can start to explain better. So it was a great opportunity for us to sort of think through how will our data stand up to some of the traditional ways that people study and do statistical analysis. And we're very excited to have the opportunity to start doing some of that work. The FDA is very interested in starting to work with us on those kinds of testing of methods within our data. And that pilot was just one way of starting to sort of open the door a little bit. So I think we have an opportunity that we haven't had before. It's sort of an unprecedented opportunity to see all of these disparate data sources and start to think how they are alike or how are they different, and then what are testing our assumptions about what that means.
And thank you. And Marcus talked about being able to distinguish whether different populations are different. But Troy, there's also just the more nuts and bolts question of you're bringing data sets from different sources together. Purportedly, they are the same types of data, but there's work to be done to understand if they're behaving the same way. Can you say a bit about that?
Yeah, well, there are a couple of components to that. You know, there has been speculation that the common data model, which was used, could potentially have an impact. We're actually conducting some work now, which may show that the source of the common data model may not create differences in the outcomes. But there are different data sets that, you know, depending upon the specific population, that have been shown to yield one result when a different population is used that would yield another, which is why designing the methods and applying those methods to the appropriate data sets are so important in ultimately getting us to the right answer.
That's great. I was at a presentation on an unrelated topic the other day where they said you can now sequence a genome for $1,000, but it'll cost you $1 million to find out what it means. Which is good. It's the same here, right? We have all this data. But it's work to make sense of it. And one of the things that we haven't talked about is so we've been talking about the scientific pieces, but there's lots and lots of work to be done on the sort of legal agreements and the ownership and the sharing and the governance. Which any big data project, you never sort of read about it, but any big data project has to work through that. Would you just say a bit about how IMEDS has been tackling those sorts of things?
Yeah, so within IMEDS, we have multiple tiers of the way in which we're governed. We have a scientific advisory committee, of which Sally, of course, is one of the members, and that committee is primarily responsible for scientific oversight of the program, of all aspects of the IMEDS program. We then have the IMEDS steering committee, of which Marcus is the chair. That is really more programmatic oversight and ensuring that the direction that IMEDS is taking is the intended direction as we set out upon this mission of ours. And then we also are governed, of course, ultimately by the Reagan-Udall Foundation Board of Directors, of which we have a subcommittee of that board, which is responsible for guiding the IMEDS program and our legal and fiduciary responsibilities. So those are really the three key areas in which we're governed.
Great. And I want to save time for a couple of questions from the room if we have any. But I should mention that in your meeting materials, Patrizia Cavazzoni from Pfizer was to be part of this panel, and Pfizer has been one of the companies that has been very committed to this effort. She couldn't make it at the last minute, but I wanted to acknowledge that we had intended to have her here. And also to use that to segue, I'm going to ask you a question that I would have asked her. But so you mentioned IMEDS evaluation, which is beginning to ask some questions of the data. Now I know that some of what we're doing is contractually secret, but could you give us a general sense of the kind of question you might begin to ask with IMEDS evaluation?
Yeah, I think there are multiple types of questions that could be responded to through the IMEDS evaluation program. First of all, a pharmaceutical company, a manufacturer may believe that there is a safety issue with one of their own products. Drug X is believed to cause hepatic failure or renal failure. And with IMEDS evaluation, they would be able to go into the system and query, again, using the same methods, the same tools, the same data sets that FDA would be using within MMHS and Sentinel, to get a better understanding as to whether or not this is a true safety signal that needs to be further investigated. Another way could be if FDA through Sentinel detects a safety issue or a potential safety issue, IMEDS evaluation will give the pharmaceutical company the opportunity to go in and better understand what FDA is looking at so that there can be a closer collaboration to understanding whether or not a safety issue actually exists. At the end of the day, what we're hoping to accomplish is to improve patients' lives and save patients' lives. That's at the forefront of every stakeholder group that's represented, certainly at this table and beyond. So by having these two programs working together, we believe that that puts us one step closer to accomplishing that goal.
And Sally, let me wrap up with you. You've thought a lot about the role of patients in contributing to data, but you've also been thinking about how we communicate findings and information back to patients. So where should we be going on this as things like IMEDS evaluation begin to produce results?
You know, it's really important to start engaging people, real people, in the experiences that we're learning about, but also they can put that into context for themselves. So I think we have to find ways of being able to give back. Think about it, for the most part, all the data we're using originates with a person, right? And behind every bit of data is a person's story. And they're the ones who have sort of the context and nuance to be able to put some perspective on it. So we absolutely have to find ways of being able to communicate back effectively what's the relevance to me or people like me. Are there ways within which we can display data that would actually be more meaningful to someone, especially at the point of care with their provider? How is this influencing the decisions that we're going to make? But I think also that it's time for us to think about engaging people more actively and proactively in the study of the data. That maybe what we need to be doing is opening up some opportunities for not just the research world to be looking at the data, but for patients and real people to start helping us look at this. As I said earlier, I think one of the things that can help us test our assumptions is the veracity that real people bring to the conversation. Marcus mentioned care guidelines. We did an experiment a couple of years ago where we just took the epilepsy guidelines that are traditionally used in epilepsy care with the American Academy of Neurology, requested us to ask our patients, how close is this to the care that you actually receive? Well, we actually saw quite a diversity of adherence to those guidelines, and a lot of it was depending on where you got your care. So it wasn't so much that people were doing necessarily a bad job, but it was really more about understanding, in my situation, in the real world, I get my care from the primary care doc, who isn't necessarily a specialist in epilepsy. What do we need to do to help them better understand what are the needs that that patient population needs? And one of the things that they were missing on was a drug-related issue. So women of childbearing age using contraceptives who were also simultaneously taking seizure meds. That's a problem. And so for the patients themselves, they weren't necessarily aware of it, but the providers should have been. And we need to find a way of being able to ensure that that kind of information is communicated effectively. So I think there's a variety of ways, but I think we need to engage people more effectively, let them know what the relevance is to them. Right now, I think all of this would probably seem rather distant from their real-world experiences, so we need to pull that in and better understand how we can communicate that.
Great. Do we have questions from the floor? Would anyone like to ask a question of the panel? Sir.
Hello, I'm Dr. Darrell Roberts. I'm actually a new employee at Battelle Memorial Institute and have been working in healthcare quality since about 2000. One of the areas of big interest to me, especially in big data, is around the development of the learning health system and how the contributions of each of these independent groups of big data can come together to form something that I, as a nurse at the bedside, because I've been a nurse for 25 years, could say, okay, I have something new here that this patient presented to me that's a question about X, and I want to know more about it now. I want to be able to answer his question now. Oh, and by the way, I want to know it for me, because someday that question's going to happen again, and I'd like to contribute my additional knowledge that I gained from this patient, this physician, this pharmacist, back into the learning health system in a useful way. How can IMEDS, Sentinel, and the people at this panel help make that a reality?
Great question, and in the interest of time, I'm just going to go to Marcus on that because it's somewhat related to your opening comment about the data gaps we still need to fill and the vision of where we go.
Yeah, everyone, I think what the first thing that came to mind when you were describing your question was the Dartmouth Institute, the variations in care. And I was in a discussion, and I did a panel at CMTP in Baltimore last week, or week before last, and I was talking to Brian Luce and Newell McElwee from Pfizer at a break, and we were talking about this issue about the variations in care, and what we really got to on this is it's really not the variations in care that are the problem. It's actually what you just said, and that is that it's our lack of ability to study those variations and the outcomes associated with them, so that we get the information, and then once we begin to learn, we communicate that back down. So that N of 1 is really an important aspect to this. I think we're trying to get there. I think as we get deeper data, as Bob alluded to before, I think use thicker data was the term. We often use the term deeper data, but it means the same thing. The deeper the data we get, or the thicker the data we get, and the better we get at understanding it, and understanding why it's there and the quality of it, I think that's actually when we're going to be able to get to the information that you're talking about. So we've got an initiative in California called CalIndex that was just set up last year, where we're trying to create sort of a next generation health information exchange by integrating data from different healthcare institutions, payer and provider alike, to create an integrated view. And the idea is to do that two-way exchange of information. So you pull it out, you can study populations better, but you can also now power decisions much more effectively. I still think we have a long ways to go because the gaps, there's a lot of leaps ahead in terms of faith to get to that point, but that's actually really where we're headed. I think that was a great example of what we want to try to get to.
Thank you. Gentleman here in the second row.
I have a follow-up on what Marcus mentioned as the part of the method is improve the quality of the data. I'm a researcher at National Institutes of Health, and I'm a heavy user of the IMEDS Cloud Lab. And I actually have kind of interesting ideas along these lines, how to improve the quality of the data. What is undecided whether I should now direct it to the IMEDS evaluation, which might have better data, or even can I expect updates to the IMEDS methods data set? To provide some examples of the things I found is when you actually look at the GE data set, I found out that it's 31 million patients, 500,000 patients in that data set had events prior birth, and I kind of emailed the ETL person saying, well, that's a problem. Should I trust the data? Another problem is in terms of quality. When we looked at, I mean, the 141 million in the CCAE data set looks very impressive. Only 6 million of that have a lab value. So when you go by claims and you look at, we had an idea about levothyroxine patients who we needed their TSH value. So you see that 43% got TSH value from the claims.
Let me stop you there. Those are really useful examples. So let me go to either Marcus or Troy for a quick response on data quality.
Yeah, it's the end of the question pretty much.
Good. And then and then to wrap up, also for people who are already using the data or researchers who may want to be part of IMEDS, what do they do?
They contact Susan Gruber. And we have a process that we go through. We need to ensure that the work that's being performed meets the requirements that we have, the obligations that we have to the data providers. Assuming those are valid, then we'll open you up to have access to the IMEDS Research Laboratory. And you can go on to the Reagan-Udall site, the IMEDS section of the Reagan-Udall site, to see specifically which researchers are actively in the lab, and I can tell you they're from a diverse cross-section of stakeholders, and we certainly welcome any and all comers to join us in our quest for developing better methods. Yes?
And Marcus, just really quickly, the issue that was raised with data quality.
Yeah, data quality is an issue, and it's the repurposing of the data for something different. And I think it shows the importance of, ultimately, when we really get to validating what we see in the data, we can use some types of data for hypothesis generation or for testing of methods, but when it really comes to validating things, when we see that, we really need to be able to get to source systems of data. Where did it really come from, and why was that, those dates, why were they transposed? Were they transposed? Was it an artifact in the data from data entry? Was it an artifact in how it was manipulated over time? Or is it something in how I ran my query on the data? So there's a quality improvement process that really it ends if all you've got is sort of an insulated database, but is really critical to get to the deeper data. You have to have ties back to the data, to the sources of the data, and that's why we have complementary types of data sets. We have the databases that we have within the lab, but we also have the ability to get to other types of data partners and work directly with them to get back deeper in the data, and that's where the complementary nature of the environments exist.
The platform is excellent. I have nothing
Yep.
good to say. It's the sense of that it's a living platform. There are kind of quarterly updates or yearly updates, and we can contribute to those. That would make the platform even better.
Thank you. So let me say that I think all these folks are here all day, or maybe not, but certainly the conversation can continue. I know there are more questions, and we want to be engaged with the stakeholder community, but in the interest of, I know there are more questions, but in the interest of keeping us to time, I'm going to wrap this up and thank the panel for a really interesting conversation.
Thank you.